So this is a continuation from the last video. Now for this video, we are going to be looking at finish up specimen B, because I started specimen A, which is granite pod with the seeds. And obviously I told you they're going to ask you to dissect or open that pod and maybe draw the lateral view. And I told you that if you're asked to draw, you should be able to know how to title, get your magnification, work with the range given. If you are given a title or if you're asked to draw a specimen, from the information or from the question where they asked you to draw this specimen, you know how to raise your title. So how do I mean? For example, you're asked to open up the granite pod and draw the lateral view of the specimen, or open up specimen A, because I won't call the name, they'll just identify it as specimen A, and draw the lateral view of what you have opened up. Now, because they said lateral view, your diagram, the title of your diagram should be the lateral view of specimen A in brackets a granite pod, close the bracket, then times your magnification. You just do times 2 times 1 times any number based on what you've calculated. Now, if you are asked to, okay, I'll go into drawing magnification later, but I think for now, let me just exhaust all the possible questions and give you the answers so that you can at least, because this exam today is what? I think today is June 12th. You have, I think the exam is June 16th. I would have made this video earlier, but I've been busy lately, so I'm sorry about that. So let's look at possible questions. Later I'll go into the drawing and how to get magnification. Possible questions you could get from specimen A has been discussed in the earlier video. Now I want to dive right into possible questions for specimen B, C, D, and the rest. For specimen B, they asked your teacher or your school to provide you with roasted granules or milled granules. What does it mean? It's more like what we call in Igbo, we call it ose oji, osoji. Or for some others, it might be granite paste, the one we used to eat bread. It can be cocoa or peanut butter rather. But whenever you crush or mill granules, the possible question they want to ask you from that is what we call a food test, particularly a fat and oil test. Now there are two types of fat and oil tests. The first one we call a translucent paper test, and the second one is what we call a Sudan 3 solution test. But this crushed, anytime you crush a food substance that has oil, and we know that the seed of a granite has oil, and they've asked us to roast and bring roasted granite seed, it means they want to ask you to carry out the translucent paper test. So they might ask your teacher to provide you with a paper, and on that paper you'll be asked to place the crushed or milled granules. When you place it on it, you will notice that later, after a while, that paper will become oily. If you now raise that paper over a light source or over where the sun is coming in, or a light source, you notice that paper, you would notice that light can pass through but you cannot see through. So such observation, you might be asked to table out your observation, maybe in the table where you may be given. The table might be divided into 3. You might have the first column of the table as food test, the second column of the table might be observation, the third column of the table might be inference. If they ask you to write your findings on that table, the first table where you have food test, you should write the test carried out is what we call translucent paper test for oil or for fat and oil. Then under observation, you now write, you observe that when you place the crushed or the roasted granules on the paper, you notice that after a while, you raised the paper over a light source, and you notice that it became translucent because light can pass through but you cannot see through. Don't say it became transparent, it's wrong. If you say it became transparent, it means light can pass through and you can as well see through. Do you understand? So use the word. From the name of the test, you also get the answer. Observation is that it became translucent, light can pass through. Then your final inference, which is the last column, your inference would be fat and oil is present. Positive test for fat and oil. Do you understand what I said? Fat and oil is present, full stop. Still under that same inference table, you write again positive test for fat and oil. So this is how you always table it out, and that's the possible food test they want you to carry out. Then you could also ask other questions relating to the disease, the placentation. Do well to check my previous video. I've already covered everything on groundnuts. Now we're diving right into specimen C and specimen D. Specimen C is water in the beaker, only water, while specimen D is water and salt, that is salt solution. And what are the possible questions? Because there's another part where they ask your teacher, it's not part of the specimen, but your teacher was asked to provide the following reagents. Iodine was there. I think copper 2 sulfate solution was there. Now, because there's iodine is a yellowish brown what's it called, reagent that they use in starch. Is a reagent they use in food test for anything that contains starch. Examples of starch. Starch is a polysaccharide, is a complex sugar. Example, rice water is starch, yam has starch, bread has starch, biscuit has starch. But they didn't ask us to provide any of these that has starch, except one, maize grain. But that maize grain, your school rather was asked to provide a dry maize grain. So they may ask you to probably crush it if they asked for mortar. But if they didn't ask for mortar, they may even ask you to drop the iodine on the maize grain and observe. That kind of test, if you ask to report it, is under the test table. You write test for iodine, test for starch, not those tests for starch, iodine, test for starch. Under the observation table, you tell them it's going to turn blue-black, that maize grain. If it has access, you must open it up so that you can have access to the maize grain powder inside the maize grain. Then the next one is the inference, which is starch is present, positive test for starch. You get observation is it turned blue-black. The color change to blue-black. So that's where I feel they can use that iodine they gave you. Another place they can use iodine is in specimen D and C. Now they could also want to carry out what they call osmotic osmosis. Osmosis has to do with movement of maybe diffusion. You get when an ink diffuses in a liquid, you drop iodine and you notice how the iodine color changes the whole water. You get that is also diffusion in liquid. They could also ask you to carry out that test with water. Another thing they could ask you to carry out the test is maybe the porosity of specimen E, which is a beaker filled with sandy soil. Sandy soil, you must know the properties. One of the properties is that it has large, it is coarse and has it doesn't have fine particles. It's coarse, it's rough. It has the particles are a bit bigger they're not fine, and they could be about 0.01 millimeter in size. Another thing is that they are porous, meaning that water easily passes through because of the air spaces between the particles. There are spaces between them because of their size. Then if you pour water into that sandy soil, you notice that the water does not stay long on the soil, it goes down. So it's highly porous. Another thing is that the sandy soil is not rich in humus. Humus means decayed substances, so it's not rich, it's not good for planting. So these are things they could ask you to use the water to do. Another thing they could ask you to use the water to do is that they could ask you to use the water in terms of the food test. Maybe if they ask you, because they ask you to get evaporated milk, and they ask your teacher to provide you with copper 2 sulfate and caustic soda, what we call sodium hydroxide. Now, these are the two reagents we use for protein tests. Sodium hydroxide, we call caustic soda, and copper 2 sulfate solution, which is blue in color, dark blue. Now, we use these two for protein tests. The particular protein test we use it for is burette test for protein, whereby you're going to have, if your evaporated milk is there, then they would want you to test it. Now, if you get your evaporated milk and you're asked to pour it into a beaker, when you pour it, and the next thing they'll ask is to add sodium hydroxide, which is caustic soda. Or if the name is not given, maybe they ask the teacher to label it as specimen X or L. If you add sodium hydroxide, what it does is that it makes the milk, it doesn't change, the color change remains the same. That's what you write. But it makes the environment more what's it called? More you can use the word more. It makes the environment easy for the color change to be observed when you add copper 2 sulfate solution. You could also make it more guys, the word acidic. You get more basic rather, because sodium hydroxide is a base, so more basic for the copper 2 sulfate to act properly. So when you now add copper 2 sulfate solution, what it does is that it changes it from the color of the milk to what? It gradually changes it to purple or violet. Again, and that is a positive test for protein. So you must add sodium hydroxide first before you add copper 2 sulfate to get your color. When you're adding that, do well to add, if they're giving you, if you're giving a particular volume or drop, do well to follow that, because you need more of that sodium hydroxide for the copper sulfate solution to change the milk into purple or violet. So these are areas you could use that water. Then we're going into the next one, specimen. I've talked about specimen E, which is sandy soil. Told you possible things they could ask from there. They would probably not want to carry out capillarity, but if they ask you what is the capillarity of sandy soil, just know that sandy soil, its water goes up, you get, through sandy soil, but it's because it has spaces for the water to easily move. Then another thing they'll ask you is the specimen F, alimentary canal of the small mammal. If your teacher provides a small mammal, you should know that the alimentary canal of a mammal, starting from the mouth to the esophagus to the stomach, you need to know the kind of food that's digested in these areas. Now, what is an alimentary canal? An alimentary canal is a pathway that food passes while it's being broken down. It's one of the parts of the digestive system. It's not the digestive system. Students tend to mix it up. Now, the alimentary canal is a part of the digestive system. The digestive system is a bigger picture where you have all organs that are part of the system, like the liver is there, the pancreas, but the food does not pass through the liver and pancreas. So the alimentary canal is any parts that the food passes through. You get, but some other part like liver and pancreas, it doesn't pass through, but they secrete enzymes that will aid in digestion. So that is why they could be part of the digestive system and not part of the alimentary canal. So, but they asked your school to provide alimentary canal that has the part from the esophagus to the stomach. So you should be able to tell them the food that is being broken down in the mouth, in the esophagus, and in the stomach. Now, food does not really, no digestion occurs in the esophagus. It's just from the mouth where you have digestion of starch. So if that mammal eats any starchy food, digestion of starch occurs there, and it's aided by the enzyme thiolin produced by the salivary gland in the mouth. Then food is swallowed through the esophagus, and the esophagus takes the food or carries the food from the mouth to the pharynx to the larynx. It passes through the larynx, but not entering. Then it gets to the esophagus. On the esophagus, it's there's towards the junction there where do you have the airway. There's an epiglottis that prevents food from going into the lungs. So it continues the movements to the esophagus to the what we call the from the esophagus to what we call the stomach. Now the stomach is where we have the protein digestion going on. Only protein is digested in the stomach, just like only starch is digested in the mouth. So the stomach, there are some things in the stomach that aids digestion of protein. The first thing you must tell them, if they ask you what kind of digestion occurs there, you say what food is being broken down, protein. What are the things it has to enable proteinous food to be digested? First of all, it has the HCl acid. What's the function of the HCl? The HCl ensures that the environment is acidic to prevent bacterial growth or any kind of decay that will happen in the stomach of the animal. And then it makes the environment acidic for proteinous enzymes like renin that act on milk and pepsin that act on other protein to effectively work. So it makes the environment or the medium, it makes the environment or the stomach more acidic for these enzymes to work. Then the basic thing you should also know is the food from the stomach moves to where? The next part is going to move to is the first part of the small intestine, which you call the duodenum. From the duodenum to the jejunum, and from the jejunum to the ileum, where absorption of food occurs. But because they didn't ask your teacher to get the small intestine or the intestine, they might not ask questions from there. All right. They might also ask you, why is it a mammal? Is a mammal? Where was this intestine gotten from? You tell them it was gotten from a mammal. Reason being that, of course, you could cite the size. It's a bit large. Also the structure. Those of mammals tend to have their large stomach size, and then they don't tend to have the crop like you see in birds and apes. Then you should also, the next one, specimen G, where you have the twig or branch of a plant with the misoto, which is a parasitic plant. Now I want to say something. When you look at this picture of a misoto, it's a plant, but why is it parasitic? It does not, like other plants that should have their roots in the soil, it has the roots in the what host, the another plant that is the host. So what it does is that it has a special root we call haustorium, Haustorium. Haustorium is a special root that mistletoe has. That's a parasitic plant called mistletoe has. It uses that haustorium to penetrate the vascular bundle where we have the xylem and the phloem of the host. So when it penetrates, it gets mineral and water, and then it uses these materials it has gotten from the host to now carry out what photosynthetic food production. So it does not get food. Don't get it wrong. Don't say that the mistletoe gets food from the host. No, you only get materials needed to what carry out photosynthesis from the host. And what are these materials? It uses what does it use to get nutrient from the host? Tell them it uses the haustorium. And what nutrient does it get? It gets water from the xylem, and it gets minerals too. Then you can also get other nutrients from the phloem. It now uses it to carry out photosynthetic food production or photosynthesis. Do you understand? Then where is it found? Always on the host. It looks like a Christmas. Can see it looks like sometimes you can see you have some spike coming out like a Christmas tree. What you see on the Christmas tree. Then the next one is J, fresh fish. I'll tackle that, but most of the time they'll ask you the habitat of the fish. What kind of fish? If your teacher provides mackerel, this one we see most of the times that is shiny. The body is shiny and glows and is a bit oily when you cook it. That's mackerel. You can tell them the habitat of mackerel is a marine habitat. But if it's a tilapia fish, the habitat of tilapia fish can be marine and freshwater. Then if it's a bony fish, depending on the fish, tell them features of the fish. It has the upper column to cover the gills, the fin for movement. It has a streamlined body. Streamlined body means that the mouth, the ends where the head and the tail towards the ends of the fish are narrow, than the center part of the body is wide, and that's for using movements in water. So streamlined body for using movements in water. And there are also some of the features it has. Streamlined body is one of them for using movements in water. Upper column or gill cover to cover the gills. It also has eyes for vision, lateral lines for detecting vibration. And what does that mean? If another organism is swimming past a fish, or another organism is coming from behind, the water vibrates, and the fish rather picks this vibration and can use it to hide from the predator if it's an enemy. So the lateral lines for detection of vibration. Are you following? All right, let's go to specimen K. Is a dry maize plant. What's a maize? A dry maize grain. Identify it as a maize grain. Is a fruit also. What kind of fruit? Is a Caryopsis. Caryopis. And why is it that? Because the pericarp fuses with the what's it called? I think the pericarp fuses with there's a fruit wall fuses with the inner part. So it's a Caryopsis. It's under true, simple, dry, indehiscent fruits. Yes. Then another thing could be a specimen LMN, where we have dry tomato paste, M, fresh tomato that is firm, that is strong, and then N, rotten tomato. For the rotten tomato, let me start. They'll actually know when you get fresh tomato, if you leave it, if it starts getting rotten, there's a white thing, that's white lines you see on those things that a rotten tomato has. Let me ask you what that thing is. Is a fungi, and it's a fungal infection that is decaying it. What kind of fungi? Is a white mold. It happens and it decays it and turns it gradually into making it more watery. And then if bacterial infection comes in, it starts smelling. Then they could ask you what can be done to prevent this state of this rotten tomato. Preservation method like freezing, drying. We could also have sun drying, boiling and blending, and then turning it into specimen K, which is a paste. They could ask you, how did you turn it into specimen K, which is a paste? Do well to tell them that you should know the procedure in turning it into a paste. You'd some persons sun dry, some persons boil and crush. They remove the water, they strain it, they dry it, the water is, and then they add preservatives, and then they can or they seal it up in a sachet. So try to know all these things. Then PQR. I'm trying to make this video short. PQR, P is thermometer, Q is quadrant. You can see it in the picture. Sorry, P is thermometer, Q is meter rule, and R, the last one, is quadrant. Now, what's a thermometer? It's just a device to carry out the or know the, what's it called, the average temperature of a body or an object. Could be telling you that it's hot or cold. Where is it used? It can be used in ecological studies. You could also use it in biology. Ecological studies to know the temperature of the environment, temperature of an object. It can also be used in ecological study. What topic in biology uses it? It is the topic we call population studies under ecology. We use it. Also, three of them, the class where they use them, three of them, thermometer, quadrant, and meter rule, are used in this topic in biology we call population studies, or rather you tell them ecological studies, under which you have the topic, the subtopic population studies. It is used for meter rule. It is used to measure an object, and know the size. It's also used to measure the length of a farmland or grassland during population studies on plant species. You want to carry out the number of plant species, that's population counting. You need to measure out an area of land. You use it. They might ask you, how do you combine quadrant and meter rule? You do that during population studies. You have to first of all map an area, then you use a meter rule to measure out the size of that area, the length and breadth. Then you throw your quadrant. Quadrant, you can see in this picture, is an instrument used to count the plant species on a piece of land. It's made up of a metal or wooden frame inside which has a particular measurement. They could ask you to know, calculate the measurement of the one provided. You try to measure it. Some schools can have 15 by 15 inches, depending. It can be 1 by 1 meter. Just know the range or the size. When you throw it, how do you use it? You throw it in the land, a mapped out area. You choose an area, you measure with a meter rule the area of that land. You choose the sampling method you want to use. In most cases, you use sampling method, sampling technique, or what we call, you can use what we call, is this method we use. You choose an area and you sampling size, more like you sample an area, and sampling technique or sampling size, and then you throw the quadrants. The quadrants, you throw it first through, you count the number of plants you have. You can have sunflower, 3 days grass, spare grass. You count them, you take, and you write first toss or first throw. You counted 2 3 days, 1 sunflower, 1 spare grass. You throw it again. We're still within that area you mapped out, that area of land. You toss or throw it again. You count the number of grasses that appeared inside those small boxes. You note their number. You toss it like 4 or 5 times, after which you take the total number of plants you counted and the total toss or throw you did. So if you throw it 5 times, it will be like total toss will be like first throw, second throw, fourth and fifth throw. For the first throw, you count the total plants you saw there. You can say you counted 2 rows, 2 sunflower, 2 3 dags. At the end of the day, total the number of rows and 3 dags from all your toss. You get your average number of what plant species. You can also use it to calculate the population density and population size, depending on the formula you're using. All right, at this junction, I want to say thank you so much. Yes, we're back again. Thank you for staying tuned up to this moment. I advise you to watch through. I'll also try to post a video on diagrams. I'll try and make a time before the exam, which is on today's Friday, which is next week, Wednesday or Thursday, I think. I'll try to do that, but also get tips, so you can put on the comment section any question you have and any confusion. I'll do well to answer. Well, to always share this channel with your friends, and let's hit up to subscribe. Let's hit up to 1,000 or 2,000 followers. I would really appreciate it. Thank you so much, and bye.